All right, thank you all for listening in. I would like to particularly thank the organizers and the society for making this meeting possible despite the difficult circumstances. We are interested in cerebellar cognitive function, and we are trying to find out how the cerebellum communicates with the cerebral cortex. We think that that is the key, and we're studying that in mice. Nonetheless, I want to start with the human brain. So here is a picture of a human brain on the left, showing the vast differences in volume between the cerebral cortex and the cerebellum, and the coronal section on the right with the white matter stained dark here shows the vast differences between the two structures in terms of white matter. The cerebral cortex has a lot of recurrent loops with cortical areas communicating with other cortical areas, whereas in the cerebellum, it's mostly an in and out fibers. There's not much going on in terms of intracerebellar loops. But the two structures are massively interconnected. And we know quite a bit about the anatomy of those connections. And this is the current and also, or if you wish, the traditional view of how the two structures communicate. So you have a cerebellar output that goes, cerebellar cortical output that goes to the deep cerebellar nuclei, to the thalamus, and to a cortical area. And then that cortical area projects back to the cerebellum where its cerebellar input originated via the pontine nuclei. And so this connection is traditionally believed to be a closed loop, one-to-one interaction between the cerebellum and the cerebral cortex. And by the end of the talk, I hope I can convince you to change your view of this communication to a slightly more complicated pattern where the cerebellum does not communicate one-on-one with the cerebral cortical structure, but rather has a role in coordinating how cerebral cortical structures communicate with each other. So cerebellar connections with the cerebral cortex are plentiful, and we know from studies using fMRI, like the study from Randy Buckner that is quite widely known, that when you look at functional interaction or functional connectivity based on correlation of neuronal activity, you find that the largest proportion of cerebellar cortical surface is actually connected with association cortical areas. So giving, more credence to the belief that the cerebellum is actually massively influencing cognitive function. But what we still don't know is, of course, what is the neuronal mechanism? So we feel that cerebral-cerebellar interaction is crucial, but what actually happens in that interaction in terms of spikes, in terms of action potential? So if you want to study cognition in mice, you don't have as many options as you have with the humans, but one option is working memory. And so we looked into the literature to find out what was known about cerebellar involvement in working memory. And this is an example from a paper by Desmond et al., where they used verbal working memory in humans and in an fMRI setting. And they identified a activity hotspot in lobulus simplex and Crus 1 when subjects had to solve a verbal working memory task. So that's a good start. So we can now look in mice in a working memory task, and we can ask if Crus 1 or lobulus simplex are involved in that. And of course, verbal memory doesn't work in mice, but mice can be tested for their spatial working memory task. And they, of course, have spatial memory, and spatial ability is quite important for their natural survival. And you can test that in a very simple assay, which is shown here. This is a plus maze that the mouse sits in and can actually freely explore. The situation that is depicted here shows the mouse after it has already explored the arms A, D, and B, and now it's back to the center. And a healthy mouse would have a tendency to avoid the arms that it has just recently visited. It would remember those two arms and would now turn preferentially into arm C. So if you follow the movements of the mouse as we would do, and we do that with a video camera, of course, tracking which way the mouse is going. You end up with a sequence of arm visits like down here. And if that sequence has 4 letters that are not repeating, so 4 arm visits without repetition, we call that a spontaneous alternation. So that's the, and of course, these sequences go on and on and on. And so when you analyze the entire sequence from 10 minutes of exploration, you can calculate what percentage of your sequences of 4 are actually without repetition, are actually spontaneous alternations. And then you get, in a healthy mouse, to percentages between 30 and 50. So between 30 and 50% are spontaneous alternations. And in this case, in the 4-arm maze, the chance level is at about 22. Now, previous studies have shown that in this kind of task where the mouse or the rat has to make a decision in a maze, there are 2 structures that play a key role, and those are the medial prefrontal cortex and the dorsal hippocampus. So here is a slightly different maze, and showing a mouse in a situation where it is acting on a learned task, and this is a delayed non-matched to sample task. So the mouse is coming here to the decision point and has to remember, my previous trial, I turned right, now the correct response is turning left, and it has to make that decision. And in a publication here by Bencinane et al., and there are other labs that have shown very similar things, they have measured neuronal activity in the medial prefrontal cortex and dorsal hippocampus, and they analyzed the coherence of oscillations between these 2 structures. So coherence of oscillations is a measure of phase stability, of stable phase relationship between 2 oscillations. And I will say a little bit more about that, why that is relevant functionally or neuronally. So the phase relationship, the stability of coherence, or the coherence value here, was highest when the mouse was at this decision point of the Y maze. So what you see here is the outline of the Y maze, but the color code gives the magnitude of coherence while the mouse is walking around in the maze. And you see that there's a clear spike right at the point where the mouse has to make a decision. So somehow the decision making requires these 2 structures to have high coherence, and high coherence, based on work that was presented by Pascal Fries starting in 2005, and there's now a vast amount of literature. High coherence has an influence on how well 2 structures communicate. If you have oscillations, neuronal oscillations in the local field potentials that are properly aligned and stably aligned over time, and that makes for high coherence, then these 2 structures align in time their time of highest excitability. Typically in the local field potential, when it becomes negative, that is an indicator of high excitability in the structure. So when both of these structures are in the negative phase of their oscillation and they exchange spike activity, the chances are increased that that spike activity will actually elicit a response in the target area. If the phase relationship is unstable or it's actually the opposite, then that will hamper neuronal communication. So coherence of oscillations is a meaningful measure, and it informs us about how well 2 structures are communicating with each other. Now, we repeated the experiments in the plus maze, now recording prefrontal cortex and hippocampus, and we basically found the same thing that Bencinane et al. Found. And I have to add that they analyzed the coherence at the moment of decision making, and they also separated the coherence values between correct and incorrect decisions. And what they found was that when the rat made a correct decision, the coherence values reached higher magnitudes than during incorrect decisions. So we did the same thing. So we had the mouse explore the maze, and then we asked, what is the coherence value? And in this case, we're focusing on gamma coherence here, 80 to 90 hertz, during the time of decision making. Now, if you look at the time axis down here, time 0 is the moment where the decision making is completed. So the mouse has now at time 0 completely left the center. All 4 paws are in the new arm. And so everything is done. The mouse has decided is in the new arm. So everything that occurs before that, these 2 seconds before, we consider that the process of decision making while the mouse is in the center. So you see here the coherence, time resolved coherence in gamma during the decision making when the decision was correct. And this is what it looks like when the decision actually was incorrect. So you see there's quite a bit of difference, replicating basically the finding of Benchimani and of others, that correct decisions have higher coherence values than incorrect decisions. And so knowing that now, even in the spontaneous alternations, is not a learned task where we get the same kind of coherence value changes in prefrontal cortex and hippocampus. The question is, how does the cerebellum come in here, right? So knowing that lobulus simplex and Crus 1 are the likely candidates that are influencing this based on the previous human data, we started out asking which one of those structures actually communicates with the medial prefrontal cortex and dorsal hippocampus, and particularly based on neuronal oscillations. As coherence is important, we were interested in are oscillations or aspects of oscillations represented in one of those or both of those cerebellar structures in Purkinje cell activity. So what we did is we recorded in all 3 structures. So we had a head-fixed mouse preparation, 2 electrodes, one electrode was inserted in the prefrontal cortex and the hippocampus. And here we focused on local field potential because we were interested in coherence of oscillations. And in the cerebellum, we focused on Purkinje cell spiking activity. So we have spiking activity data from the cerebellum, Crus 1 and lobulus simplex, and local field potential oscillations in hippocampus and prefrontal cortex. So analyzing that data, we did first, we just looked at the instantaneous phase of oscillation. So we separated the local field potential, broadband local field potential by the typical frequency bands, delta, theta, beta, gamma, low gamma, high gamma, and then asked, if I take the instantaneous phase of each of these frequency bands, of each of these oscillations, and ask if my Purkinje cell in the cerebellum represents or is in any way interested in that phase, what's the answer? So here on the left, you see a rate modulation of a Purkinje cell in the lobulus simplex that's modulated by the instantaneous phase of delta oscillations in the medial prefrontal cortex. So we found a lot of these cells, and what these plots on the right show is the fraction of Purkinje cells that actually represent instantaneous phase in the medial prefrontal cortex or the hippocampus for each one of these frequency bands. So what you see here for lobulus simplex, there are two peaks stand out. So there's a lot of Purkinje cells that represent instantaneous phase in delta oscillations, and a lot of Purkinje cells represent instantaneous phase in high gamma. And that's true for medial prefrontal and dorsal hippocampus. Now, if we go to Purkinje cells in Crus 1, the picture changes a bit. We still have lots of Purkinje cells that represent instantaneous phase in delta, but no representation, or very few that represent high gamma. So there's quite a difference here. And so after we saw this, we've assumed, okay, lobulus simplex is probably where we should look in terms of gamma involvement in the coherence of prefrontal cortex and hippocampus. But so instantaneous phase is only one part of the story. Coherence is about phase difference, stable phase difference. So the next thing we did is we asked, how do Purkinje cells represent phase difference, if at all? So this little schematic here shows an imaginary Purkinje cell that is interested in phase differences of oscillations between hippocampus and prefrontal cortex And this particular Purkinje cell here really likes a phase difference of 270 degrees. It fires at a very high rate, and it really doesn't like much 90 degrees, fires at a very low rate. So this is the idea, to basically find out what the firing modulation is, if any, in relationship to phase differences between prefrontal cortical and hippocampal oscillations. And what you see here is an example Purkinje cell that beautifully shows beautiful modulation with phase difference of these oscillations. And this is in a gamma oscillations, and you can quantify that modulation using circular statistics, and you get a resultant magnitude vector that gives you a quantitative value that you can then further analyze. And that's what I'm showing you here. These are now results. Each dot here is a Purkinje cell. And what we did is we used these resultant vectors and we expressed them as R values, which is basically a z-score equivalent measure expressing the magnitude of those vectors relative to the randomized control. And so anything that's above 1 is significant. So to our surprise, we saw that the frequency differences, the phase differences, are equally well represented, first of all, in both structures. So both Crus 1 and lobular simplex represent phase differences, and they're equally well represented in all frequency bands. So there's a difference between phase representation and phase difference representation. So a lot more Purkinje cells actually represent phase differences, and that is broadband through all frequency bands, from delta to high gamma. Now, knowing that now, the question becomes, does that mean that the cerebellum might actually have something to do with modulating the coherence in the prefrontal cortex and hippocampus? And so, first of all, we wanted to know what happens when we stimulate the cerebellum, the lobulus simplex in particular, to the behavior. So we put the mouse back in the plus maze, and we had an LED placed over lobulus simplex, and the Purkinje cells were expressing channel rhodopsin. So we could stimulate Purkinje cells, reduce DCN output optogenetically, and we did that at the moment of decision making, or at the beginning of the decision making process. So these experiments were done by Yu in the lab, and he basically observed the mouse through the video system. And when the mouse was approaching the center and stuck the nose into the center, he pressed the button and started a 1-second optical stimulation of the Purkinje cells in lobulus simplex. And then we compared these stimulated trials with non-stimulated trials, and the result was quite dramatic. So in the control trials, the mouse had its typical performance around 40%. But in the stimulated trials, the performance dropped to chance level. So the mouse lost the ability to make correct spatial working memory decisions. So the behavior was clearly dramatically influenced by the cerebellar stimulation. What happens to coherence? So these graphs here basically show you a summary of the coherence time course that I showed in these pseudo color plots earlier. Now this is basically a sum over all the frequencies from 80 to 90 hertz, and it shows the time course of coherence during the process of decision making. Again, time 0, sorry, is the moment the mouse has left the center. And you see that in the correct decision in green versus the incorrect decisions in red, coherence reaches far higher values, significantly higher values in the control trials when the decision was correct. Now, when we stimulate the cerebellum, that difference disappears. So there's no longer a modulation of coherence that is reflecting decision outcome. So now we have two elements to this result. So we have the behavioral outcome that is clearly affected. The cerebellar stimulation drops spatial work memory performance to chance level, and the modulation of coherence that is so clearly associated with the decision-making process disappears. So, of course, we would like to think that the influence on coherence is causally related to the decision outcome, but that statement, of course, we cannot make. But the idea or the suggestions I have for you now is that what the cerebellum actually is doing in its communication with the cerebral cortex, rather than just talking to one structure in a closed-looped way, that the cerebellum actually modulates coherence between cerebral cortical structures in a task-dependent manner. So now the projections from the cerebellum to the cerebral cortex actually get a new meaning, that they're not there to influence activity in individual structures, but it's there to influence the communication strengths by modulating coherence between two structures. So the cerebellum becomes a coordinator of functional connectivity in a task-dependent manner. So, and this is findings, of course, that we made based on our cognitive task on spatial work memory, but we're actually not the first to report an influence of cerebellum on coherence. This was actually first reported by Daniela Popa and her colleagues. They recorded baseline coherence in a rat, in this case, I think, and they were recording in primary sensory and motor cortex and infused mucimol into the interpost nuclei, basically silencing cerebellar output from the interpost. And what they found was a dramatic drop in gamma coherence between M1 and S1 after inactivation of the interpost nuclei. So clear indication that cerebellar output is required to maintain normal coherence between these two structures and translate that to functional connectivity, to communication. Here again, an indicator that this is not just in association cortical areas, but also applies to motor areas. And this is a very recent paper, very similar finding, but now in a behavioral context. This is from Lindemann et al. Came out in PNAS just a month or two ago. And they found that when you provide an air puff to the whiskers, there is an increase in gamma coherence between M1 and S1, these structures representing or related to the whisker barrels. And when Purkinje cells are stimulated, thus with chamberdopsin inhibiting cerebral output, it results again in a significant drop in gamma coherence. So the picture now becomes that the cerebellum is acting as a coordinator of rhythms, of neuronal rhythms in this case. But can that be, expanded? Is that a general principle? And I would suggest it is, because a lot of rhythms are motor rhythms, right? So there's most essential movements are actually rhythmic, like chewing, breathing, walking, licking, swimming. And so besides the rhythms of the brain, we have all these rhythms of the body. And this is likely that the cerebellum has a role in coordinating these rhythms as well. And so there's not much directed study in coordination of these rhythms by the cerebellum yet, but there are some indications. So this is from a publication from my own lab from 2013, where we recorded in the medial cerebellar nucleus. And so we found that medial cerebellar nuclear neurons, they project massively to the brainstem, and they project to areas that have the respiratory pattern generators, the whisking pattern generators. And so we were wondering what the reason or what the function of that might be. And we recorded neuronal activity in the medial cerebellar nucleus, and we found that many of these nuclei, actually the majority of the nuclear neurons, represent rhythmic movements, and many represent more than one. We actually found even 2 in our sample of, I forgot, 24, that represented all 3 of these: breathing, whisking, and fluid licking. So that's quite interesting, and they're projecting to the brainstem, so they might be involved in coordinating these movements. And you might wonder why breathing and whisking are coordinated, but we know they are. But breathing and fluid leaking, they have to be coordinated, because when you drink water and you take in water, you have to swallow it, and then you have to stop breathing. So there's a reason why that happens. And so that brings me to the end. So we think that the role of the cerebellum is more like, or that coordinating rhythms is an important role of the cerebellum that translates into cerebral-cerebellar interaction, into the coordination of neuronal communication by coherence. And in this last slide here, I want to thank all the people that did all the work. So Yu and Stu McAfee, they did most of the data recording and data analysis. Roy Silito is an old friend and colleague. He provided the mice, ataxic mice, his mouse model that he created by eliminating Purkinje cell synaptic transmission. Brittany Correa, Mia Fox, and Angela Taylor contributed as graduate students in the lab, and Benjamin Smith is currently our lab technician. And of course, I want to importantly thank all people that provide us the money that allows us to do this. NIH, of course, National Institute of Mental Health in particular, and the Neuroscience Institute, which of the University of Tennessee, which provides us always with great support for our graduate students. Thank you.